Okay, so what I hope you see is that enzymes are of critical importance because they basically make metabolism happen the way it should. They're the difference between, just a heap of sugars and fats and things like that, and a functioning metabolism. And every cell requires thousands of them. Every cell requires thousands of enzymes, and every enzyme has to have the right shape. And if it doesn't, it won't work. Do is assemble amino acids over and over and over again into many thousands of different chains that will always fold up to have just the right shape. Many reactions are driven by a set of enzymes, which again you can compare to machines on an assembly line. I'll give you a fun example. And again, this has to do with alcohol. If you drink ethyl alcohol, that's the drinkable type, it passes into your bloodstream and does various things that may or may not involve dancing on tables with a lampshade on your head or whatever it is. Not that I ever did that. And your blood ultimately flows through your liver, and your liver is what breaks down a lot of toxins. And one of the enzymes that your liver cells make is called alcohol dehydrogenase. That takes ethanol. Ethanol fits into that little active site, and it converts it into a compound called acetaldehyde. Acetaldehyde itself is still toxic. If you have too much acetaldehyde in your body, it causes headaches, flushing of your skin, nausea, other symptoms like that. In fact, acetaldehyde is responsible for many of the symptoms of a hangover. However, acetaldehyde is broken down by a second enzyme called aldehyde oxidase, and that breaks acetaldehyde down into acetic acid, which is much less toxic. You eat acetic acid. Well, I don't know how often you do, but people do. It's the active ingredient in vinegar. So you might eat it in salad dressings or things like that. And acetic acid is pretty easy to excrete. So it's a two-step process. You turn ethanol into acetaldehyde using alcohol dehydrogenase, and you turn acetaldehyde into acetic acid using a second enzyme, aldehyde oxidase. Here's a mushroom. It's called Coprinus atramentarius. It's actually edible. People do eat it, although it doesn't store. You have to eat it very freshly picked. The problem is that there's a chemical in Coprinus that blocks aldehyde oxidase. So think back. We go from alcohol to acetaldehyde to acetic acid. If you block that second step by blocking aldehyde oxidase, then the alcohol turns into acetaldehyde, and the acetaldehyde doesn't have anywhere to go because there's no aldehyde oxidase to break it down any further. The upshot is like Lucille Ball trying to package chocolates. Sorry, old TV reference if you don't know. Acetaldehyde builds up in the body, and the result is the hangover from hell. So you can eat these, but you cannot serve wine with the meal or for, I think, up to 36 hours after eating these mushrooms. Because if you ingest any alcohol, even a little before eating the mushrooms or up to 36 hours after, the alcohol will be broken into acetaldehyde, but that second step, breaking down acetaldehyde into acetic acid, that second step won't happen. You build up acetaldehyde and you end up with nausea, headache, flush skin. You end up with a hangover. In fact, there are various drugs that do the same thing. This is one of them, disulfiram, sold under the trade name Antabuse. and it's used to treat alcoholism. It won't stop you from drinking, but because it blocks your aldehyde oxidase, it makes you deathly ill before you've even drunk enough alcohol to feel drunk. You get hit with a terrible hangover as soon as you start drinking before you've even got much of a buzz on. So it definitely discourages you from drinking, and drugs like this are in fact used to treat alcoholism. So there are drugs that can block specific enzymes in a metabolic pathway, in a series of enzymes that do something. Now, going back to the early 1900s, a British doctor by the name of Archibald Garrod was studying a genetic disease called alkaptonuria. If you have alkaptonuria, you produce urine that looks normal when it comes out of you, but it contains high levels of a compound called homogentisic acid. And what you need to know about homogentisic acid is when it's exposed to oxygen in the air, it turns black. So your urine will look like normal urine when it comes out. Let it sit for about 30 minutes and it looks like freaking ink. So yeah, on the right, that's the urine of someone with alkaptonuria, where the urine's been sitting out for 30 minutes. It's worse than that. It's not just that your pee turns black. Homogentisic acid is produced in the body, and a human can excrete most of it, but they can't get all of it. And if you have alkaptonuria, homogentisic acid will start building up in your skin, your eyes, your heart, and especially in cartilage. There is a person with alkaptonuria whose ear is looking a very odd shade of blackish blue. That's because homogentisic acid is building up in the skin and also in the cartilage. Homogentisic acid in your cartilage causes joint damage as well, and people with alkaptonuria develop very severe arthritis, usually at a pretty early age. They'll get arthritis starting in their 30s or so. Okay, why do you get this? Homogentisic acid is produced when cells break down one of your amino acids, an amino acid called tyrosine. Tyrosine is one of the 20. You need tyrosine to make proteins. Most people get much more tyrosine than they need in their diets. So the excess is broken down and disposed of. So tyrosine is actually broken down through several steps. We don't need to worry about what they are. Through several steps, it breaks down into homogentisic acid. That is normally broken down into a form called 4-mallyl acetoacetate, and then that gets broken down further. Ultimately, you can burn it as a source of energy. So I'm not showing a lot of those steps, but there's lots of them, and they all depend on a different enzyme. And the one we're going to focus on here is the step that turns homogentisic acid into 4-mallyl acetoacetate. But if there's one enzyme called HGD, stands for homogentisate dioxygenase, doesn't matter, don't memorize these names, know that this is a case study of a more general problem. If you're making faulty or you're not making HGD enzyme, then that pathway is blocked, and homogentisic acid can't be converted to mallyloacetoacetate. It builds up in the body. Most of it is excreted by your kidneys, which is why your pee turns black. But some of it builds up in your cartilage, including your joints, which is why you get severe arthritis. Okay, so the absence of a particular enzyme causes the symptoms of a particular disease. And the reason this matters is that Garrod studied families with a history of alkaptonuria, and he did a pedigree analysis, just like you're supposed to do in Lab 5. And he discovered that alkaptonuria is inherited by simple Mendelian rules. It's a simple Mendelian recessive allele. So here we've got an enzyme that you're not producing. The inability to make that specific enzyme is inherited as a simple Mendelian trait. And from studying this and other human genetic diseases, he actually studied 4. He concluded that what a gene physically was the instructions for making one enzyme. As he put it, one gene, one enzyme. So a gene is the instructions for making an enzyme. There is a gene that enables you to make the HGD enzyme that turns homogentisic acid into formylylacetoacetate. If you have 2 copies of the normal gene, you make the right enzyme and you're normal. If you have 1 copy of the normal gene and 1 copy of a faulty gene, the normal gene can still make enough HGD that you lead a normal life. But if you inherit 2 copies of a faulty allele for HGD, if you inherit 2 copies of a gene that contains the wrong instructions for making HGD, that means that you can't make the enzyme and homogentisic acid will build up in your body because you've got 2 copies of the instructions for making HGD and both of them are faulty. Both of them make an enzyme that doesn't work. So now we're finally getting to where the pieces on the jigsaw puzzle are starting to meet. We've been talking about what genes were. Now we finally have some idea that genes are what enable you to make specific enzymes. And by the way, we can expand this now. What a gene is the instructions for making one polypeptide, one protein. And incidentally, this is a bit of a callback. This is a guy with alcohol intolerance on the left before he's had a drink and on the right 15 minutes after he's had a drink. I talked earlier about you have this enzyme called aldehyde oxidase that breaks down acetaldehyde into acetic acid, right? You have this enzyme that breaks acetaldehyde down. If you block that enzyme by eating the wrong mushrooms or taking a certain drug, then you build up acetaldehyde in your body and drinking alcohol makes you feel bad. There are some people that are born with alleles for aldehyde oxidase that are ineffective or less effective. And they naturally show alcohol intolerance. They always have a bad reaction, even to small amounts of alcohol. For some reason that for our purposes is not important, is it tends to be most common among Asians, although I don't believe it's restricted to them. So you can block aldehyde oxidase by taking a drug or eating Coprinus atramentarius mushrooms, but you can also be born with instructions for making a less effective form of aldehyde oxidase. You can be born with their aldehyde oxidase partially blocked. And if that's the case, then you always have a bad reaction to alcohol. And this is what this unfortunate person has. I'll give you another example. This thing that looks like, psychedelic spaghetti is a diagram of an enzyme called phenylalanine hydroxylase. And what it normally does is convert an amino acid in your food called phenylalanine to a different amino acid called tyrosine. And then, by the way, the tyrosine that you don't use gets broken down into, among other things, homogentisic acid, which we've already talked about. So phenylalanine hydroxylase converts phenylalanine into tyrosine. Most people get more than enough phenylalanine. So most people, phenylalanine deficiency is not a problem. Most people have more than they can use. If you have a faulty gene for phenylalanine hydroxylase, if you can't make this protein with this shape in exactly the right way, then you start building up excess phenylalanine because you can't break it down or convert it to tyrosine or anything else. The reason this matters is that phenylalanine in too great a quantity is toxic to the nervous system. So if phenylalanine builds up in the body and you can't do anything with it, it builds up until it starts poisoning your nervous system cells. And if this is not caught early, you end up with severe mental retardation. Fortunately, it's a very easy test. All newborns are tested for phenylalanine hydroxylase deficiency. It's basically a matter of adding a dropper full of chemicals to the first wet diaper and seeing if there's a color change or not. It's very easy to diagnose with a urine test. And one thing most newborns have no difficulty with is producing urine, let me tell you. Anyway, so you can test for this very early, and there's no cure, but you can lead a normal life. But what you must do is avoid food with high levels of phenylalanine. And you can actually see this on bottles and cans of Diet Coke. NutraSweet or aspartame is about half phenylalanine. You might remember a couple of lectures back that I showed you how aspartame was 2 amino acids stuck together. One of them is phenylalanine. And so people with phenylketonuria, people who can't make this enzyme, must not eat large amounts of phenylalanine or they will suffer the neurotoxic effects. And so this is why you have warnings on labels of anything that contains NutraSweet. People with PKU must not drink this stuff. Okay, there we go. So finally, what I hope is happening in your minds is that the jigsaw pieces are coming together. We started with Mendel and talked about his peak plants and flower colors and things like that. So, and he established that there are such a things as genes. Studies on chromosomes and ultimately the work of Thomas Hunt Morgan and his students established that whatever genes are, they are physically located on chromosomes. Watson and Crick and Ross Franklin established that and lots of other people established that what chromosomes are made of is DNA, and that DNA has a particular shape, and it's made of nucleotides that include the bases adenine, guanine, thymosin, and cytosine, and it's got the double helix shape and everything. Okay, so there we are. We've just covered proteins, and it turns out that what a gene is the instructions for making one particular protein. That protein isn't always an enzyme. It doesn't have to be. But in the examples of alkaptonuria and PKU, it is an enzyme. You can inherit faulty instructions for assembling any one particular enzyme. Okay, whoa, you with me now? So we can expand Garrod's insight more in that a gene is responsible for making one polypeptide chain of amino acids. It's not necessarily an enzyme. The collagen proteins in your skin, or the fibrillin proteins in your extracellular matrix, or the keratin proteins in your hair are proteins that aren't enzymes. They do other things than that. But a gene is still responsible for making those. Every gene is responsible for making one amino acid chain. For those amino acid chains to work, they have to fold into the right shape. They've got to have the right primary, secondary, tertiary, quaternary structure. That structure depends on getting amino acids in the right order. If you don't put amino acids in the right order, you won't get the right shape. Ultimately, the reason why you get PKU is because you can't put amino acids together in the right shape so that they'll fold to give you the enzyme phenylalanine hydroxylase. And that runs, by the way, in families as a simple Mendelian trait. So you can inherit either normal or non-functional alleles of the gene for phenylalanine hydroxylase. Those enable you to make either normal or non-functional forms of the enzyme. And if you have two copies of wrong instructions for making that enzyme, then you have PKU and you can't break down your phenylalanine. Okay, so hopefully this is the point where all of the pieces have to come together and we're finally in sight of the top of the mountain I've been trying to get us to climb. And it turns out that the instructions for putting amino acids together in the right order are encoded in the order of DNA bases. This is the genetic code. Remember that DNA is made of bases that contain adenine, thymine, guanine, and cytosine, or AGTC, AGTC. In the early days, it was thought that maybe DNA was just a repeat, that every strand of DNA was just AGTC, 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 or something like that over and over and over again. Turns out it ain't so, but it's not random either. The order of bases as you go down a strand of DNA is actually the code that tells a cell how to put amino acids together in the right order. And we call this the genetic code. Okay, where's this happen? Think about eukaryotes for just a moment. Your DNA is kept in the nucleus of your cells. Normally, it does not leave there. If the nucleus breaks open and the DNA comes out, something has gone horribly wrong and the cell is probably about to die. Yet we actually now know that proteins aren't made in the nucleus, they're made outside the nucleus. So how does the information get from the nucleus to the outside? Soon after discovering DNA structure, Francis Crick proposed what he called the adapter hypothesis, which is the hypothesis that genetic information is copied onto a second molecule that shuttles out of the nucleus. The example I always use is, let's say you like to cook at home, and you're always looking for new recipes. Pretend for a moment the internet's not there. Let's say you're up in the library and you find an old cookbook that has an interesting recipe that you want to try. However, let us say that you can't take the book out of the library. Maybe it's on 2-hour hold. Maybe it's a reference book. Maybe you have not paid your fines. Come on, Wagoner. For whatever reason, you can't take the actual book out of the library. What you would probably do is just make a copy of the recipe and take that out of the library and take it to your home kitchen where you could actually make the dish. That's the way this works. DNA is like that cookbook in the library that can't leave the library. You could take the information from that cookbook, copy it onto a little piece of paper or something, and carry that information out of the library to the kitchen where you're actually going to cook the food. Same thing goes on in cells. That molecule that shuttles out of the nucleus is a second type of nucleic acid, and it's called RNA, ribonucleic acid. Ribonucleic acid is like DNA in that it's a polymer of nucleotides. However, RNA is single-stranded. It's not double-stranded, not normally. I think you could make double-stranded DNA, and sometimes you'll have a single-stranded piece of RNA that will bond with itself and form kind of a hairpin loop. But you can think of RNA as being normally single-stranded. It doesn't necessarily form a helix. I mean, you could probably make it do that, but it doesn't usually form a helix. RNA nucleotides contain the sugar ribose instead of deoxyribose. And RNA doesn't use thymine. It uses a slightly different base called uracil, which we abbreviate with the letter U. And then finally, RNA will actually go out of the nucleus of a healthy eukaryotic cell. So here we can compare, on the left, that's a DNA nucleotide with the sugar deoxyribose and the base thymine. There's an RNA nucleotide on the right that contains the sugar ribose. See, the ribose has an extra oxygen group stuck to it, which means it's ribose. If it were missing that oxygen, it would be deoxyribose. Get it? So ribose on the right, and the base that's sticking up there looks a lot like thymidine or thymine, but it's actually called uracil. Okay. We're in the nucleus. Take a length of DNA and it begins to unzip. Now, we mentioned that this happens when DNA is being replicated, right? When you copy DNA, that double helix unzips, and then new nucleotides come to make complementary strands on each of the unzipped strands of DNA. That's DNA replication. Transcription looks like this, except that in this case, the cell assembles RNA nucleotides to make a complementary strand to one of those DNA strands. This follows the old familiar pairing rules. ATGC on a strand of DNA bonds with UACG on the RNA strand. So if I have a strand of DNA that spells GATTACA, right? Gattaca, that was a movie title a while back. GATTACA, then the strand of RNA that matches with that, well, G will pair with C, A will pair with U, because this is RNA we're talking about. T will pair with A, T will pair with A will pair with U, C will pair with G, and A will pair with U. So Gattaca on DNA will become CUAAUGU. CUAAUGU. Quaugu. Yeah, that could be the title of the sequel to Gattaca. It would be Quaugu. Right. When that RNA strand is completed, it peels off and the two DNA strands come back together. And that RNA strand is called messenger RNA, or mRNA. That's the copy of the information that's kept on the DNA. And there's roughly what it looks like. There's an enzyme that does the actual job of assembling RNA nucleotides into a strand of RNA. The nucleotides are, for your purposes, they're always pretty much floating around. And the DNA unzips, and RNA polymerase brings those nucleotides in and welds them into a strand that's complementary to the DNA strand. So then that messenger RNA peels off and leaves, and that messenger strand is carrying the information that was present on the DNA strand. So this messenger RNA is analogous to the copy of the recipe that you made when you were inside the library. The messenger RNA strand passes out of the nucleus if it's in a eukaryotic cell. In prokaryotes, of course, there is no nucleus and everything just happens in the same area, more or less. And the messenger RNA goes out to a tiny structure inside the cell called a ribosome. And there's not just one, there are probably tens of thousands of ribosomes in a typical cell, maybe more. A ribosome is roughly 20 billionths of a meter long. That's what? Okay, 1 micron would be a millionth of a meter. So we're talking, what, 0.002 microns long. That's way the hell too small to see with a light microscope. We do have better microscopes now that can get some detail on what ribosomes look like, but you'll never see this with the ordinary type of light microscope that we use in most of our classes here. Each ribosome itself is made up of about 50 proteins and also four molecules of another type of RNA that we don't need to get into. Ribosomes are made of proteins and a special type of structural RNA. But for our purposes, it doesn't really matter what it's made of. What matters is that the messenger RNA slips into a groove on the ribosome. The messenger RNA makes like Madonna in 1983 and gets into the groove. Oh, you got to prove your love to me. Yeah. Sorry. Still got the high notes, man. Okay, right. That's a model of what a ribosome would look like if you can see it. Don't worry about the details. It looks like a couple of heaps of spaghetti, I know. What you need to see is that some of it is shaded in kind of bluish, some of it is shaded in gray, and in between the blue and the gray subunits of a ribosome, there is a groove. And the messenger RNA can slide right into that groove. Okay. There's another type of RNA that you need to know about called transfer RNA. And a transfer RNA molecule is one strand, but parts of the strand will actually form bonds with other parts of the strand, and the molecule twists on itself to form a set of loops. These are usually diagrammed as looking a little bit like, cloverleaf highways or something like that. There's several little loops that stick out. One of these loops is twisted in such a way that it's got 3 base pairs that stick out in a prominent position, like 3 little, buck teeth sticking out. That loop is called the anticodon loop. Okay, the other end of the tRNA is bound to an amino acid, and thanks to some magic that we call aminoacyl transferases, and you can think of as little fairies, which amino acid is present depends on what bases are present in the anticodon loop. So here's a simplified molecule of what one of these looks like. It's a single strand shown in yellow. There are several places where the strand forms complementary base pairs with itself, which is what locks it into this kind of cloverleaf shape. And at one end of it, you have 3 base pairs that stick out, called the anticodon loop, down there below. At the other end, you have some bases that are bonded to an amino acid. And which amino acid it is depends on what bases are present on that anticodon loop. The order of nucleotides in the messenger RNA determines the order of amino acids in the protein. The nucleotides are read 3 at a time, and a grouping of 3 nucleotides is called a codon. There are 64 possible codons. Remember, this is messenger RNA, so you could have AAA, AAU, AAG, AAC, AUA, AUU, AUG, AUC, ... I'm not going to tell you all of them. Every codon is associated with 1 amino acid. Now, there are 64 codons and 20 amino acids used to make proteins, So most amino acids are associated with more than one codon. But the exception is 3 of the codons are called stop codons. They don't code for an amino acid. They don't mean, hey, this amino acid goes here. They mean stop assembling the protein, we're done. Like the period at the end of a sentence. They stop this process. So here's a ribosome with a groove on it on the left. And on the right, here's a piece of messenger RNA that, like old Flat Top, comes grooving up slowly. Get it? That's a Beatles song. Here come old messenger RNA, he come grooving up slowly. He slips into the groove on the ribosome, and he makes like Madonna, gets into the groove. Oh, you... Okay, sorry. Sorry, it's just me in the house. I'm probably starting to crack up. So the messenger RNA slides into the groove on the ribosome. At its beginning, there are some nucleotides called a leader sequence that we don't need to worry about. But once that beginning has slipped in, you have 3 nucleotides fitting into the ribosome, and they're AUG right here. Everybody look at the AUG that are right on the ribosome. Here come the transfer RNAs, and there are several transfer RNAs that will all try to fit into the slot. There's a little slot in that groove on the ribosome, and they'll all try to bond with the messenger RNA, but the only transfer RNA that can fit is the one that has UAC on the anticodon loop. Right? You could think of this as, again, think of it like a bunch of keys in a lock. Many keys might be shoved into a lock, but the only one that will fit is the one that has teeth that fit the lock tumblers. Many transfer RNAs might try to fit into that slot, but the only one that can do it is the one that has UAC on its anticodon loop, because that's complementary to AUG on the messenger RNA. Now, thanks to the work of some, they're not really fairies, some specialized enzymes called aminoacyl transferases, the transfer RNA that has UAC on the anticodon loop always carries the amino acid methionine. So the transfer RNA comes in, its anticodon loop is complementary with those first 3 bases on the messenger RNA, and it's always carrying the amino acid methionine. Now, the messenger RNA and the attached transfer RNA goes ka-chunk, 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 and moves down 3 spaces. So the messenger RNA moves 3 nucleotides down, and now the messenger RNA's nucleotides UUU are in that slot. And many transfer RNAs will try to fit, but the only transfer RNA that will fit is the one that's got AAA on the anticodon loop. And the only transfer RNA that is packing AAA on the anticodon loop is the one that is carrying the amino acid phenylalanine. Okay, so the AUG on the messenger RNA binds with the transfer RNA that has UAC on the anticodon loop, and that one brings in methionine. The next 3 are UUU, and those bond with the transfer RNA that's got AAA on the anticodon loop, and that one brings in phenylalanine. So the only transfer RNA that matches UUU is the one with AAA, and that one is always carrying the amino acid phenylalanine. Now a peptide bond forms between the 2 amino acids, and that first transfer RNA falls off. And it's ultimately recycled. We don't have to worry about what happens to it. Now the ribosome goes ka-chunk, 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 and 3 new nucleotides are brought into that slot, and you can see that they are UCA We are one of a few universities in America whose initials are also a messenger RNA codon. I mean, there's University of Alaska at Anchorage would be UAA, and University of Georgia is UGA. And of course, one of our rivals in the Southland Conference is Abilene Christian, and they're also a codon, ACU. I guess since college and university begin with C and U, those are both RNA nucleotides, so there's probably a number of universities that are also RNA codons. But here, we've brought in the three next nucleotides, UCA. Those can only bond with a transfer RNA that's got AGU, and the transfer RNAs that bring AGU in on the anticodon loop are also the ones that are carrying the amino acid serine, S-E-R, and so on. What will happen now is that the transfer RNA with AAA will fall off, and the ribosome will go click, 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 and the next three nucleotides will come in. They spell out UAU. The only transfer RNA that can fit will be the one that's carrying AUA, and I don't remember what amino acid that has, but the process will just keep repeating. And the complete code book looks like this. Each one of those three-letter abbreviations stands for one of the 20 protein-forming amino acids. So UUU is phenylalanine, UCA is serine, UAU would be tyrosine, CCC would be proline, lysine is the Canadian amino acid because its codon is A. Get it? Canadians supposedly say A all the time. So lysine is a Canadian amino acid because its codon is A. Never mind. Okay. And then I guess proline would be the Spanish amino acid because its codon is C. Okay, never mind. I thought it was funny. Okay, right. Once the ribosome hits a stop codon, the process comes to an end and the completed protein falls off. The ribosome separates into subunits. And then that protein may have to be modified chemically before you can use it. And we call that post-translational modification. We don't really need to get into that too much. Okay, right. This is a lot to absorb. And it may take more than one viewing of this to get it all in your head, and it may take some more work. I'm going to go ahead, it being 11:02, I'm going to give you guys another pretty generous break. What the heck, we'll come back at 11:20 and we'll do something else. So I'm going to go ahead and pause recording. Takes a minute for the for my and here we are. We will stop the recording.